Okay, welcome back everybody. I think most of the people are in now. I did warn you that if I was going to have another chance of telling us how wonderful you are and what fantastic things you're working on at the moment. So Dilek here has a microphone. So is anybody here wants to tell us something about what they're up to or project partners they're looking for or capabilities they have on offer to potential partners or anything else interesting that's happening in their organisation or their country? Come on, here we go. Here's a brave lady here. Thank you.
Thank you very much. Hello, my name is Agnieszka Tabernacka and I represent Department of Biology at Faculty of Environmental Engineering at Warsaw University of Technology. And I must admit that our department didn't participate as a partner in any ERA-net project yet, but actually we are not exactly industrial biotechnology. We are mostly involved in environmental engineering in field of environmental protection. But as we know, every product of every technology finally ends up in the environment as a waste or wastewater or something like that. And therefore, what we can offer is our expertise in actually in the fate of our product in the environment. And these are two different fields of interest that we can and expertise that we can offer. One thing is what will be the result of the presence of our final product or byproduct in the environment. So risk assessment. Ecotoxicological risk assessment based mostly on ecotoxicological tests, and we are specialists in this field. We can offer standard test, ecotoxicological test, as well as genotoxicity tests to test our products, our byproducts, and the effect, the impact of the product of our new technology in the environment. And the other thing is biodegradation tests and biodegradability of the byproducts of the technology, as well as the final product of the technology. So we will be very glad to assist in every project in those fields. Thank you very much.
Thank you. Very good. Very good. Okay. Anyone else? You don't have to come up, but you can if you like. Do you want to come up, Stephanie, or do you want to stay there? It's up to you. Do you want me to be your glamorous assistant and hold the brochures? There you go.
Thanks. I'm Stephanie from the Flemish department EWI. We are one of the partners of the ERA-IB team. And we are responsible for the communication of ERA-IB. And we have developed a call brochure of the results of the first joint call. And now the second joint call has finished. We ask you to also contribute to the call brochure of the call brochure of the second joint call with the results. So I don't think I have enough examples, but you can find it also on the website of ERA-IB. But you can have a look, and then you will see what is expected from you. So I will pass them around. And also a few copies of my colleague, Monica Soorman, of the Flemish study about bio-based economy in Flanders. Okay.
And just Stephanie, on the projects that have completed from the second call, we're going to probably use some of the information you've given us already that's for public dissemination, but you might ask for extra information like pictures or things like that. Yeah. Yeah, here you go. So these are going to be outside, are they? Yeah. They'll be outside. On the table outside. Okay, that's great. Okay, anybody else Theodora, you wanted to say something? Do you want to come up, or you're going to come up? Yeah. So Theodora covers the main research council, which she'll tell you.
So I'm from BBSRC, which is the main UK research council for non-medical biological research. We've just funded 13 networks in industrial biotechnology and bioenergy, which we think cover pretty much the whole landscape, from high-value chemicals from plants to bioprocessing, pharmaceuticals to systems biology of biorefining. There'll be something there for everyone. And the point of these networks is that they have networking events similar to this, where you can meet industrial collaborators, interested academics. They're not just for UK residents. We'd be really, really keen to see some international involvement in them. So if you are interested in seeing what the UK is doing, please come and talk to me, and I'll be able to give you contacts for the relevant network for your area. There's no buy-in, it's completely free to join. Not only do these networks do events where you can meet people, but there's also proof of concept funding available through them for small-scale projects to get the preliminary data and the proof of concept. So you can go on to larger projects through ERA-IB, through FP7, through your own government's funding processes. So yes, please come and talk to me about them. I'm obviously very keen to tell you about them, and they're great.
Okay, thanks, Fedora. Okay. There goes a gentleman here.
My name is Marcin Wolański. I am from University of Wrocław, and we have participated in a gene drug project. And our team works with streptomyces for years, and we have expertise in heterologous expression and gene engineering and in vitro assays, like different EMSA footprints and more complicated stuff. And we can offer this service for anybody who would like to cooperate. Thank you.
Thank you. Very good. Thank you. Okay. On at the back. Do you want to?
Okay. I'm Dominic Grant from the University of Warsaw. Although I'm a faculty of chemistry, but we are a bioinformatics group. So we do different things from bioinformatics based on sequences, but also based on structures. We are really good at comparative modeling. We can produce structures from sequence. We cannot claim that we can produce a structure for any given protein sequence, but if can be done, then I think we are the best choice to do this. And there is a biannual competition. It's a blind test called CASP that experimentalists, they hide structures into drawers and they publish just sequences. So theoreticians, they predict the structures, and then there is a conference where the two worlds, they meet, and then the results are compared. So we do pretty well on this competition. And given that our main competitors are from the US, not from Europe, then I think from European-based projects, we are pretty good partners. Thank you.
Great. Thank you. Very good. Okay. Any more brave souls? Otherwise, we'll move back to the presentations. Yeah, that's a good idea. So let's see. It's the one with 13 in it, wasn't it? Yes. Okay. Here we go. So Gerard's going to start us off on the next session, but also tell us a little about his organisation as well.
Yeah, Berns, I come from SIAP. Oh, sorry. Saxon Institute for Applied Biotechnology. So you can find information on the website www.siap-biotechnology.de, and our main activities are in the field of bioprocess engineering. We are able to carry out fermentations in our research campus up to 400 liter, including downstream processing. we have good cooperation to, for instance, CBP in Leuna, and then it is possible up to 10 cubic meter fermentation to carry out, and also, for instance, pre-treatment of lignocellulosic substrate we made in cooperation with the industry. The main activities are in the field of fermentation of industrial strains, Trichoderma and Penicillium verruculosum strains, but we have also activities in the field of biogas and plant, also in vitro cultivation of plants. Thank you. Also, wie geht es hier? And now I will present our ERA-IB project with the acronym products from lignocellulose. Sorry. Excuse me.
Sorry. There we go.
So, and here, here.
The two big
Yeah, okay. Excuse me, please. So, the objectives, as the following main tasks have been studied. The first task is the development of a pre-treatment process which allows the separation of both lignin and carbohydrate, cellulose content. Secondly, the development of an Penicillium verruculosum enzyme complex optimized for the SSF process, the simultaneous saccharification and fermentation. And the investigation of the SSF process, and finally the modification of the separated lignin for the production of fiber-reinforced biopolymers, as well as for the production of fine chemicals. Altogether, there are 7 project partners. CIAB in cooperation with the Leipzig University and also responsible for the coordination, 2 institutes of the Technical University of Dresden, Institute of Plant and Wood Chemistry, Institute of Wood and Paper Technology, the Freiberg University of Mining and Technology, the VTT in Helsinki, LNEG, a partner in Lisbon, and the main activities in lignin modification were done at a subcontractor formed by FBR, Food and Bio-Based Research in Wageningen, and finally we had a cooperation with Biotechnol in Bucharest, Romania. To the first topic, pre-treatment of lignocellulose. The main focus are the investigation of 2 different processes up to a pilot scale. Here you see the natural pulping process with formic acid and hydrogen peroxide. It is the process is illustrated here, and we have an incubation about 30 minutes at without pressure, about 100 or 103 degrees centigrade, and then we have the pulp and the lignin, or we find in the liquid phase, and it is the precipitation is with water, and the pH of 1.5 is by the formic acid. The second pre-treatment up to a pilot scale was the alkaline pulping of wheat straw with sodium hydroxide, and also separation, washing, and precipitation of the lignin in the black liquor with chlorine or with sulfuric acid. Why investigation of different methods for pretreatment? It is necessary, and also we investigated the different properties of lignin and pulp depending on different pretreatment processes, which is important also for the, for instance, in case of lignin, for the modification of lignin, and why a scale up. Also, first we had for all project partners, we had to supply enough lignin and also pulp, and also investigations how the properties are changed by a scale up. And the verification of the process scheme we did in pilot scale. So here is listed what was assayed. The assay, the analytic of pulp, the intrinsic viscosity. There had been differences between the pretreatment processes, the determination of crystallinity by X-ray diffractometry, scanning electron microscopy, the composition of pulp. This is important. How much lignin do we have in the pulp after the pretreatment process? The hollow cellulose content, and also the content of xylo-oligosaccharides, but this is in case, is it investigated in case of autohydrolysis. The autohydrolysis was done in Lisbon at the project partner LNEG in Lisbon. The lignin was assayed, the influence of pulping duration and the liquid ratio, what changes do we have in this case? The influence of formic acid in case of the natural pulping, the influence of sodium hydroxide, functional groups and molecular weight is important, glass and lignin, and also ER spectroscopy.